Now that you understand the basics behind monohybrid crosses and the two major conclusions that Mendel reached from doing those crosses with those 7 traits that he did with the pea plants, now I can take you one step further. Mendel was a scientist. He was very curious about his topic of study. So now, with those 2 conclusions that he reached about alleles and about segregation of those alleles, it opened a whole nother can of worms for him. And this led him down a path that will center around dihybrid crosses. You know how to do monohybrid crosses when we're looking at just one trait, one set of alleles. In dihybrid crosses, we're looking at 2 traits at the same time. Now, the reason he went in this direction is from one specific question that he asked himself, especially after he figured out that second conclusion that gametes, when they form, alleles have to separate. You have these traits, the alleles, the 2 different copies, they segregate out into the sperm or the eggs when you go through meiosis. And you only get 1 copy of that allele per egg and per sperm. So Mendel then had a question. Does the segregation of one set of alleles affect how another set of alleles segregates? So what he did is he stayed with his 7 same traits. He still stayed with the seed color, the shape color, the flower position, all those 7 traits that we were talking about. He stayed with those, but now he looked at 2 traits together. For example, what he did is Mendel performed crosses that followed 2 different traits. And this is what you call a dihybrid cross. So for example, one of the crosses that he could do, say we took true breeding plants, so true breeding meaning they are homozygous dominant or homozygous recessive, true breeding plants with round yellow peas, and you crossed it with a pea plant that was wrinkled and green peas. What do you think he saw at the end of this? This is your peas, your parental. What do you think he saw after his first crossing with that first familial group? When you break this down, this parent can only give big R's and big Y's. This parent can only give little r's and little y's. So what you would come up with is that the first filial generation was all heterozygous for the 2 different traits. However, their phenotype, what they actually displayed, would look like this parent right here. All of the offspring would be round and yellow. But this isn't the generation that he was interested in. He wanted to take it one step further, just like he did last time, where he goes beyond the first filial group and goes on to the second filial group. This is the one where he was curious what would happen. He already knew what was going to happen after the parental into the first filial, because you're taking a homozygous dominant and a homozygous recessive parent, you know you're going to get a heterozygous offspring. So in order to really test this, he had to go beyond that and go to the F2 generation. When he did that, what do you think he saw? In order to figure this out, what Mendel actually ended up doing is he took the F1 generation and he crossed them with each other. So he took a heterozygous and crossed it with another heterozygous. Now, these Punnett squares work largely the same way as the monohybrid crosses that you've been doing, the Punnett squares for them. However, before you can actually do this, one thing you have to do is you have to discover all of your different gametes, all of the possibilities. I will walk you through how to do a dihybrid cross in class, but for this one, I want you to try to figure out all of the different gamete possibilities that would result out of this, just like when we did a monohybrid cross. Say we were just doing a cross like this. I know that out of this one, I can give a big R and a little r. I can give a big R and a little r. So that my Punnett square, when I worked it out, would have big R, little r, and then you'd have your your big R, little r. Just like how you did it with this, you got to figure out all of your possible gametes first for this type of cross, and then we will work through this in class. And I will tell you what the conclusion was from these types of cross, and then we will practice more of the dihybrids when you get to class. So I will see you there.